Mechanisms of infectious disease, mostly just as an introduction. So we'll talk about the different types of infectious microorganisms and how they're structured, as well as some of the important terminology with respect to disease. So when we use the word infection, this refers to the presence or multiplication of a particular microorganism within your body. And that differs than colonization, because colonization means that a microorganism is actually established a presence long term. For instance, you would have microbial colonies on the surface of your skin, but it wouldn't be appropriate to call that an infection because it's not necessarily causing disease. It's just sort of present. Now, this one here says, do you have any infections or colonizations at this moment? List as many as you can identify. And are they normal or are they making you ill? Well, you think about where you might find microbial colonies inside a human body. They're going to be in areas like your gut, right? So you're going to have lots of microbial flora in your colon, as well as microbial flora on the surface of your skin. And then infections can be wide and varied all throughout the body. Now, colonizations can be normal, whereas infections we think about it as being abnormal and can make you ill. Now, microbial flora or microflora refers to all of the microorganisms that are living in or on your body. Most of these are useful and typically don't have any effect on your body, but some of these can be pathogenic. And what's interesting then is that certain organisms that might live inside of your body, like let's say E. coli, that's part of your normal microbial flora, but under the right conditions, they can cause disease. They can become pathogenic. So most of these can actually cause disease if your health and immunity are weakened, and we say that in that case they're opportunistic, right? Like you can actually get a staphylococcus infection in skin if your skin surface barrier has been weakened, or if you're immunocompromised, you're more likely to get a staph infection that way. And so some of these can be opportunistic. Now, true or false, all interactions between humans and microorganisms are detrimental. Well, we know that that's actually false, right? Because some microorganisms perform important functions in your body. For instance, in your gut, there are microbes that help synthesize vitamins for you. Like certain B vitamins and vitamin K are actually produced by gut bacteria. It's also true that microorganisms help prevent infection because they can outcompete sort of pathogenic bacteria. Think about like the good bacteria in your gut. They're not going to cause disease under normal circumstances, and they actually outcompete the pathogenic bacteria that might otherwise cause disease inside of you. So the first type of pathogenic, we wouldn't consider this an organism, but we do consider this a pathogen, and that's the example of a prion. So a prion stands for proteinaceous infectious particle that lacks nucleic acid, and this was actually discovered in the 80s. And initially when it was discovered, it was believed to be false, and it couldn't be that proteins could be infectious. But we found out later that actually was true. There are infectious proteins, and there's no DNA or RNA, but they are infectious because this malformed prion protein can actually convert other prion proteins to the malformed, and they actually kind of can multiply in that regard. So these abnormally shaped versions of your own proteins can cause normal proteins to also change shape and become new prions. Now, the reason why this is bad is that these abnormally shaped proteins can start to clump together, and they can cause damage to cells through inflammatory mechanisms and also the production of free radicals. Now, this can lead to degenerative diseases, like especially in the central nervous system. These prions are more intimately concentrated within your brain and spinal cord. So you're going to find a lot of neurodegenerative diseases that are caused by prions. An example of this is mad cow disease or Creutzfeldt-Jakob, right? So the prions in a cow that might be infected, if you ingest a prion from a cow, then if it's absorbed along your digestive tract, that can actually start to convert your normal prions into abnormal ones. They start to clump together in your nervous system, and that can lead to basically bovine spongiform encephalopathy, or BSE, which we also call mad cow disease. It effectively is where you end up with holes in your nervous tissue from inflammation and necrosis. Now, viruses are, an infectious microorganism, And these are similar to prions in the sense that they're made of protein. However, these contain nucleic acid, right? Like a virus is going to have DNA or RNA, and they may or may not be enveloped, but they're going to have a protein coat that encases that DNA or RNA. Some examples of these could be things like retroviruses, like HIV, where essentially this virus can insert its own genetic information into your DNA, and it becomes part of the DNA of certain cells of your body. Now, a virus we call an obligate intracellular parasite because they can't replicate without a host, and they use the host cell's metabolic machinery to make new viruses. This is why it's parasitic. And so some people think viruses are not living because they require a host cell. It really just depends on what your definition of life is. But in any regard, these viruses are, multiplicating cellular devices that essentially can spread rapidly and efficiently, and they have a high mutation rate, so these can actually evolve pretty quickly. Now, when we look at the structure of a typical virus, they can either be, sort of naked or enveloped. If they're naked, then essentially we say that they don't have an envelope, and essentially you just have this protein capsid that encases, the nucleic acid core here. Now, we call this a virion, which is one infectious particle, viral particle, and it's typically not going to just be DNA or RNA inside of this virion. Most viruses will also carry with them some kind of cellular machinery that helps to improve the infectious cycle of this virus. This could be things like a DNA or RNA polymerase. There could be other enzymes that are involved with like breaking up protein or helping to insert the DNA, viral code into a host cell. There's a whole host of enzymes that viruses can create and package with them. Now, they can also be enveloped, and an enveloped virus is one which has a phospholipid bilayer around the outside. And this is going to have these little glycoprotein spikes that are involved with basically binding to a host cell and allowing that virion to fuse with the host cell in order for the virus to become infectious. Now, it turns out that these enveloped viruses are the ones that are destroyed by alcohol. So when you think about like alcohol in antimicrobial wipe, it's actually going to punch holes in the membrane here, and it dehydrates the virus, and it makes it no longer infectious. Now, bacteria are another class of infectious microorganisms, and we consider these as prokaryotes, right? Because they don't have any kind of organelles other than the fact that they have a plasma membrane in a cell wall. These can contain RNA and DNA, and they'll have a single chromosome that's actually in the shape of a plasmid, which is a circular chromosome, and they'll have a cytoplasmic membrane, a cell wall. They can produce biofilms, which are essentially like an extracellular matrix that these bacteria can proliferate on and kind of grab onto, and they use hosts for food and shelter. Think about all the bacteria that are on the surface of your body. What's interesting is actually we have more bacterial cells in our gut than cells of our own body. If there are 100 trillion cells of the human body, we have way more bacterial cells in the gut. So it's interesting how they're tiny and they're very abundant. So bacteria come in different shapes and sizes. They can be if they're circular, then we call them a coccyx. If they're kind of rounded and elongated, we call it a bacillus. And they can also be vibrio, sort of spiral shaped. Now, this is going to relate to the sort of subtype of bacteria, and that's how we can identify them. And one subclass of bacteria we call the mycoplasmas, and these are smaller than the average bacteria. They lack cell walls, and these are also obligate intracellular parasites. So, like viruses, they're gonna live within a host cell or just near a host cell. And like bacteria, they contain RNA and DNA as well as sort of a membrane. And some examples of this could be things like the Rickettsia, the Chlamydia, or the Coxiella. And essentially, these are mycoplasmas, a subtype of bacteria that live within a host cell, and they actually use sort of the nutrients and internal environment of that host cell to proliferate and divide. Another class of infectious microorganisms are the fungi, right? These are actually eukaryotic, which means they're very similar to human cells. Their cell walls are different from bacteria, and most of these fungi require a cooler environment than human core body temperature. So a lot of fungal infections are going to be superficial or cutaneous. You think about like fungal infections of the skin, things like ringworm. That's actually due to a fungus. Now, so these are going to be more surface rather than systemic. Now, some examples of this would be things like yeast and molds. And yeast is actually really abundant on the human body. Candida albicans is abundant within the mucous membranes of your body, like oral cavity, the genitourinary tracts. And then molds are also abundant on the surface of your skin. Now, parasites include a different, sort of wide variety of microorganisms. They could be protozoan, they could be helminthic, they can even be arthropods or sort of insects. Protozoans are the unicellular organisms that produce sort of malaria as well as giardia and amoebic dysentery. And remember, these are unicellular eukaryotic organisms or protozoan. Now, the helminths are actually multicellular things like roundworms, tapeworms, and flukes. And these can live in your digestive tract or even go systemic, like the lancet liver fluke likes to live within the hepatic vasculature. And arthropods include things like ticks and mosquitoes and mites, lice and fleas. We'll talk a lot about these arthropods and how they cause sort of disease in the skin later when we get to the skin chapter. But it's important to note that parasites also include a different variety of eukaryotic organisms, not just fungi, but also protozoan, helminths, and arthropods. Now this question here asks, which pathogen is an intracellular parasite consisting of a protein coat surrounding a nucleic acid? Are they prions, viruses, bacteria, or protozoa? We know it's a virus, right? Viruses have no organized cellular structures. Because viruses can only replicate inside another host cell, prions can't reproduce at all. However, they can convert other prions to their misshapen form, right? They have this sort of abnormal conformation, and they can induce that abnormal conformation or shape on other normal prion proteins, and that's how it becomes infectious in that regard. Now, portals of entry just refer to different ways that microorganisms can get inside of a host, or us. They can come through penetration, where they might actually enter through places where the barriers of your body are disrupted. You might have this like a cut or sort of smaller wounds along the digestive or respiratory tracts. This can come from direct contact as well. Things like STIs and other congenital infections are going to occur from intimate contact. They can also occur through ingestion or inhalation. So the question here says, how many ways could you have become infected today? If you think about it. And how could you have experienced this? So like, what are some different ways you may have come in direct contact with a pathogen? Like, did you kiss anyone today? Did you inhale, the same air as someone who maybe just sneezed seconds prior? There's lots of different ways that you may have actually had a portal of entry here. Or did you ingest a pathogen or inhale one? Did you have a zoonotic exposure? For instance, you might have bats that could carry MERS, which is a type of virus that causes respiratory infection. Or did you encounter a raccoon that was carrying rabies or something? Zoonosis refers to transmission from another animal. Or did you come in contact with a nosocomial infection? Nosocomial refers to an infection that's acquired in a hospital. And there's unfortunately a lot of nosocomial infections because hospitals have a lot of people with diseases and infectious diseases. And because they're in a tightly concentrated area, you might go in for a certain treatment but end up acquiring a nosocomial or hospital-acquired infection. Or a fomite. A fomite is an inanimate object, things like a doorknob or a utensil, that could carry a pathogen. And so a virulence factor are things that make microorganisms more infectious or more likely to cause disease. Some examples of virulence factors include toxins. We have exo versus endotoxins. An exotoxin is something that a microorganism would secrete, and then an endotoxin is typically part of a cell or part of that microbe that can cause disease once the cell dies or lyses. We also have adhesion factors that help infectious organisms stick to the body. Things like the glycoprotein spikes in a virus are considered an adhesion factor. And then we can also have evasive factors, things that actually allow for a microorganism to hide or evade the immune system and proliferate within your body. And there's a whole host of evasive factors. An example of one could be where HIV actually evades the immune system because it's going to insert its own genome into the genome of your T helper cells, and it hides there in the DNA of your T helper cells. And every so often, then it can kind of replicate and resurge from certain populations of T helper cells within your bloodstream. Examples of toxins we talked about were endotoxins and exotoxins. Remember, the endotoxin is typically part of the cell. You can find that gram-negative bacteria, because they have lipopolysaccharide, or LPS, that's considered an endotoxin. And this is toxic because it leads to host immune reactions. An example of this, let's say with like an E. coli infection. You might have an infection of E. coli in the gut, and if you're given an antibiotic for that, and it kills off a lot of E. coli at once, all those dead E. coli start to release this endotoxin, lipopolysaccharide, from their cell wall, and that in itself can lead to inflammation, which can lead to shock, right? Which is actually going to cause disease there. Now, we also have exotoxins. These are usually proteins that release bacteria that can damage or kill host cells. And an example of an exotoxin could be like cholera toxin, which is secreted by Vibrio cholera, but it actually induces the cells of your gut to basically secrete more water and chloride, which actually makes your stool more watery, so it gives you diarrhea, and that aids with the sort of portal of exit and the replicative life cycle of cholera, which is interesting. But that's considered an exotoxin because it's secreted by a living cell. Now, true or false? Certain bacterial cells release proteins called endotoxins during growth. Is that true or false? Well, we know that's false, right? Because exotoxins are proteins. Endotoxins are not protein. These are going to be lipids or polysaccharides. And endotoxins are not released by living cells. These are released by dead cells. And it's interesting to think about, too, because if you're infected with a microorganism that has endotoxins, you wouldn't want to kill off this microorganism too quickly because that could, in itself, could lead to disease. And so often you want to approach infections of microbes that have endotoxins in kind of a slow pace because you don't want to release too many of these endotoxins at once. So with this scenario, we say that a woman's stomach contained bacterium Helicobacter pylori. And this is the one that causes gastric ulcers. And for many years, the woman was healthy. Then she took on a new stressful job, moved, and began care for elderly patients. A few months later, she began to suffer stomach pains and vomited blood. She was diagnosed with a bleeding ulcer. So with respect to this scenario, how do each of these terms relate to her case? What was the portal of entry for Helicobacter pylori in her gut? Well, that would have been through the digestive tract, right? She would have actually basically swallowed some of this H. pylori at some point. Maybe she shared a spoon with somebody or kissed somebody with H. pylori. It's actually an infectious microorganism that's transmissible from person to person. In fact, now that she has H. pylori, she could transmit that to somebody else, like a close loved one. Now, site-specific pathogen in this case is going to be, her gastric mucosa, and the reason being is that H. pylori is an acidophile. It's an acid-loving type of bacterium that thrives within the acidic environment of your stomach. And in this case, it's opportunistic because in her, she just took on a stressful job and also moved. Those can be some of the most stressful events in her life, up there with death in the family. And an opportunistic pathogen like H. pylori takes advantage of the fact that stress leads to a weakened immune system. And so then that H. pylori bacterium can really kind of take hold in the gastric mucosa if there's a weaker immune response. Now, H. pylori has some evasive factors because it resists phagocytosis due to the structure of its membrane. And its invasive factors occur not because of anything that's like an exotoxin or an endotoxin. H. pylori is invasive because of the fact that it causes so much inflammation, you get a loss of function and the necrosis of a local area of gastric mucosa, which leads to an ulcer, which allows that H. pylori to invade even deeper within the ulcer and get nutrients from the gastric mucosa itself. And so just some different terms that relate to infection and damage. Words that end in itis refer to inflammation, and this may or may not be due to infection. It could just be from trauma. And then emia refers to the blood. Now we can talk about things like viremia or bacteremia, which refers to those things being in the blood. So I'll give you an example, like bacteremia or bacteria in the blood can lead to meningitis or inflammation of the meninges. And we just use these roots to describe that. Now, sepsis or septicemia refers to a bloodstream infection. So if someone's gone septic, then we're talking about how bacteria, maybe from their gut, have spread into the bloodstream where they can disseminate to a lot of your internal organs. So which stage of infection are you in? If you are in the incubation stage, prodromal, acute, convalescent, or resolution? And how can you tell? Well, the incubation stage is going to be that period of time where an infectious microorganism has entered the portal of entry, wherever that might be, whether it's ingestion or respiratory or penetration. But with the incubation stage, there are no signs and symptoms. So, for instance, you may have inhaled a virus, like let's say rhinovirus, and even though that virus has started to replicate within some of your cells, that's the incubation stage because there might not be any signs or symptoms like early on while it's beginning to replicate. And then the prodromal stage is the period of time when you first get the onset of signs and symptoms, like think minor signs and symptoms, up until the acute stage. Now, the acute stage would be the most severe signs and symptoms of that disease. And then the convalescent stage means that there are no apparent signs or symptoms. And then resolution means that, that basically if there was any damage that occurred, then you've repaired that damaged tissue and you're back to normal. So the scenario here says a 5-year-old boy has an ear infection, complains of ear pain and cannot hear on that side. You see red bulging eardrum with pus behind it. He has a fever, sweats, and complains of joint aches. And then blood tests show elevated white blood cell count. So the question here is, which of these are a sign and which of these are a symptom? And we want to differentiate between these two. So a sign is like an objective observation, and that would be like the clinician's observation of a particular aspect of the patient. And then the symptom is something that the patient can feel, right? Or they might kind of talk about themselves. So ear pain would be a symptom because the patient can say, my ear hurts, right? Or the fact that they can't hear on that side could also be a symptom because they would say, oh, I have difficulty hearing out of that ear. If you see, like if you're the clinician and you see a red bulging eardrum with pus behind it, that would be a sign because that's an objective observation from the clinician. And then if he has a fever, sweats, or complains of joint aches, well, it depends, right? If the patient talks about sweating a lot or having achy joints, that would be a symptom. But if you observe the patient sweating or measure fever with a thermometer, right, then those would be signs. And then if you run some blood tests, and those blood tests show elevated white blood cell count, that would also be considered a sign because that's an objective observation that the clinician is making using that test. So in terms of diagnostic measures, like how can we determine signs? Well, you can determine a sign of a disease, using culture, serology, or detection of different blood, aspects. So a culture would just be like propagation of a microorganism. So you might do a culture of someone's tonsils to see if a certain microbe is growing there. Or you can actually perform serology, which measures basically serum antibodies in the host. And the purpose of serology is to detect things like, antibodies against certain viruses or microbes or parasites. And you can also do other forms of serology which detect antigens or certain genomic sequences or metabolites that are associated with infection. Now, after exposure of infectious agent, the body can produce antibodies. And what we're going to find then is that sometime later, depending on whether you've been exposed to this infectious agent before, antibody titer will rise. So IgM is a gigantic antibody. It's huge. And this type of antibody actually rises and falls during the acute phase. So we're going to find that IgM is the first antibody that's produced during the acute phase. And then IgM falls after the acute phase, but IgG remains elevated after the acute phase. And the reason being is that IgM, being a large antibody, can be cut up or cleaved. We call it antibody class switching, where this can be cut up into smaller antibodies like IgG. And this is going to explain why it remains elevated after the acute phase. Now, what's interesting, though, is you can look at the types of antibodies that someone's producing to determine whether they're in the acute phase or if they're in the convalescent phase. So a month-old baby is ill in this scenario. And we say that serum analysis shows that this month-old baby has IgG antibody against HIV. and then IgM against pneumocystitis. So if this individual is producing IgG against HIV, that tells us that she's already been infected with HIV for a period of time. Like she's out of the acute phase of the HIV infection. And that could have been weeks or months prior. And then IgM is going to be more indicative of being in the acute infectious stage. And in this example, they're saying it's pneumocystis. And so this would explain the fact that, okay, so she had HIV, that put her at risk for an opportunistic infection of pneumocystis. So it was HIV that came first because IgG is the older antibody, right? It's been around for a while. And then IgM is the newer one, so it means that pneumocystis came second, right? And that's sort of some inferences we can make based off of this type of serology. And now we have different drugs against bacteria and viruses, and there's actually a lot more drugs to kill bacteria than viruses. And we have more drugs to kill viruses than to eradicate prions. So the question here says, why has it been easier to develop antibacterial drugs than antiviral drugs? Well, viruses mutate really frequently. In fact, the mutation rate of a virus can be as high as like 1 in 1,000 base pairs, which means that every time a virus replicates, you got 1 in 1,000 of those base pairs is going to be different than before. And so if the viruses mutate really rapidly, that means they're changing rapidly, which means they're evolving rapidly because evolution is change. So it's difficult to develop antiviral drugs because these viruses mutate and evolve really rapidly. You might have a drug against a virus 1 year, and then the next year, if the virus is mutated enough, it might have less of an effect or no effect at all, depending on whether it's evolved against that drug or not. Now, some bacteria don't evolve as quickly. They still evolve more quickly than humans, but they don't evolve as quickly as viruses. So it's been easier to develop more stable drugs for bacteria. And if you think about the difference between a bacteria and a virus, there are a lot more things to target in a bacterium than a virus. Viruses are really simple microorganisms, and there's not that many things you can target, right? Now, bacteria are more complex, and there's a lot of things you can target. And so it's easier to find a drug that might inhibit bacterial growth because there's a lot of things that you could block to prevent growth of that bacteria. So we can't use antibacterial or antiviral drugs to destroy prions because remember, prions don't replicate. They can essentially, they can't actually infect. But they can replicate themselves, but only by modifying the existing protein in someone's body. So the only way to block a prion would actually be to prevent its effect on the normal prions in the body. So it doesn't replicate the same way as like a bacteria or a virus. So these drugs wouldn't have an effect. Now, antibiotics inhibit bacteria by either inhibiting their synthesis of their cell wall, so they're weaker and they can't survive. Certain antibiotics can target the bacterial ribosomes, so it inhibits protein synthesis. You can inhibit nucleic acid synthesis or inhibit bacterial metabolism. So lots of things to target here. But here's the problem, though, is that remember, bacteria can evolve. They change because of their mutation rate. And so we say that bacteria fight back. I mean, not literally, but eventually what we're going to find is that if you have a drug that works against a bacteria, due to evolution, eventually these bacteria will change and could resist that drug's effect. So we're saying that bacteria can fight back, not directly, but indirectly by evolution, through inactivation of antibiotics, either altering antibiotic binding sites, using different metabolic pathways, or changing cell walls to keep antibiotics out. So antivirals inhibit virus by inhibiting like viral DNA synthesis. You can inhibit viral enzymes, like we actually have reverse transcriptase inhibitors, which HIV uses to replicate. And then you can also prevent viral binding to cells, which would prevent its portal of entry. So if you can't allow it to bind and fuse, then it can't infect. And you can also inhibit the production of the protein coats or capsids. And so usually when we talk about antivirals, you're going to use a cocktail or a mixture of different drugs that act on all of these different mechanisms. That way you can really ensure that that virus isn't going to replicate. So antifungals inhibit fungi by targeting things that are unique to this type of eukaryotic cell. So remember, fungi are also eukaryotes, just like a human cell. Now this is a challenge then for us to have drugs against fungus if their own cellular biology is very similar to our own. But what we find though is that fungi have cell walls. Human cells, although they're also eukaryotic, don't. So a lot of antifungal drugs actually target cell wall synthesis. So like ergosterol in fungal cell membranes, we can use that to actually bind and form membrane holes and help to explode these fungal cells, which is pretty cool. There's also a type of sugar you find in fungal cell walls that we can target its synthesis to weaken the fungal cell wall and prevent these cells from dividing very well. So question here says, which type of infections are treated based upon the results of a gram stain? If you guys remember, gram staining is one of the types of stains we can use to identify fungus, viruses, bacteria, or parasites. Well, the example here is bacteria, right? Because gram stains actually target, whether they're gram positive or negative, and we can actually treat those infections with the knowledge that those bacteria might be gram positive or negative. That's going to tell us about their cell wall orientation. And so certain bacteria respond better to gram positive drugs or gram negative drugs. And these are actually drugs that help to target cells that are either gram positive or negative based on the structure of their cell wall. So if the cause of the bacterial infection is unknown, we can just have broad spectrum antibiotics, which basically target both gram positive and gram negative microorganisms. But if you think about this, are all bacteria bad in the body? And the answer is no, right? Remember we talked about how there's microbial flora? And so using a broad spectrum antibiotic, though, is going to even affect the good bacteria in your body. So, you want to avoid these as much as possible. This would be more of like a last resort.